I'm Anne Riddell, and this is Science in You. Our genes, they are the roadmap to how our bodies work. And with genetic research exploding, we know more almost daily about how our DNA can cause disease and potentially cure us as well. Over the next half hour, we'll explore some of the most cutting-edge developments in the field, from cancer treatments to your ancestry. But first, we start here at the New York Genome Center. Most of us march through our days rarely thinking about why our bodies do what they do. But our genetic makeup is a driving force in our lives, for good and bad. But what does that mean outside of the science lab? We went to the New York Genome Center for some answers. How would you explain in sort of the simple terms what genetics or genome research is?
It's a good question. You can I think of genetics and genomics as understanding a library of instructions. And that library really, it contains many different books and many different volumes, and it's not small, it's very large and it's complicated. And all those instructions together help make you who you are and me who I am.
Instructions, that makes sense, but then what?
And what our bodies end up doing is taking all those instructions and making the types of cells that we have. So they make bone cells and blood cells and nerve cells and muscles. And when all goes well and the library is being accessed and read correctly, then that's very good. When things don't go so well, then there are different parts and different bits and pieces of the information in that library that either get corrupted, which we call mutations, or damage to our information, to our DNA, or they go missing. Sometimes some books get checked out of the library and they don't come back. And that can have some pretty severe implications to how our bodies function.
As scientists unlock the mystery of genetics, they understand more and more about what variations can cause disease and potentially what can be done to treat them.
The field is growing astronomically fast. The technologies that have developed in the past decade allow us to not just read one strand, but to read millions to billions of them at the same time. Well, what it means is instead of it taking 10 years to sequence a human genome and costing, an astronomical amount of money, it takes a few days now. And the cost is not terribly high. If we're using this information for informing lives and health, now the turnaround isn't 10 years. As the technology continues to grow and to become smaller and less expensive and more accurate, then what I hope to have at some point is going to be a bedside sequencer where we don't have to ship off to a lab.
There are thorny questions that come with the rapidly advancing science in this area. Is more information always better? How much do we want to know?
Would I want to know if I'm going to develop a disease? That's a personal question, and I think that's one that you have to answer for yourself.
Scientists now believe cancer is a genetic disease, essentially a scrambling of the DNA in our cells. So what does that mean for patients? Dr. Max Gomez explains it could be a revolution in cancer diagnosis and treatment.
Cancer is probably the most dreaded disease of all. And now cancer researchers are coming to the realization that cancer is basically a genetic disease. A scrambling of the DNA in our cells somehow leads a normal cell to transform into a cancerous cell. Why is that understanding important? It's going to revolutionize cancer diagnosis and treatment.
My primary care physician found that I am having anemia, and there was no obvious reason for the anemia, so it's unexplained anemia.
Unfortunately, a bone marrow biopsy revealed that Mahmoud Hamid's anemia was multiple myeloma, a cancer of the blood. Radiation and chemotherapy put Mahmoud's myeloma in near remission for several years, but
gradually it started to flare up again, and it showed in the lab results.
Fortunately for Mahmoud, Mount Sinai is one of a growing number of medical centers that are doing genetic characterization of cancers, decoding the exact sequence of DNA that makes up our genes and chromosomes, something that would have been unthinkable just a few years ago.
It would have cost thousands to millions of dollars and months to just do one. Now we can do that much faster in a matter of days to weeks at a much lower cost.
What researchers have learned by sequencing the genome of many different cancers led to an unexpected treatment for Mahmoud's myeloma. More on that later. But more importantly, oncologists now realize that the cause of cancer is fundamentally genetic.
Uncontrolled growth of a cell leads to cancer. Now, what leads to uncontrolled growth? So, it is due to completely reprogramming the genetics within the cells. So, you have to scramble the genetics of an individual cell to make it to become a cancer.
The potential causes of that genetic damage, called mutations, are many. There are environmental triggers such as smoking, alcohol, air, water, and industrial toxins, and radiation, including from medical x-rays. Then some mutations are random, with many billions of cell divisions daily in the human body, each one requiring exact replication of DNA strands containing 3 billion building blocks. It's easy to see how errors that lead to cancer could eventually occur. That's partly why most cancer incidence goes up with age. Most importantly, though, knowing the genetic composition of cancers has led to a new way of classifying cancers based on their mutations rather than the organ they happen to grow in.
If you have the same mutation in a skin cancer, then it is also seen in a lung cancer. So the drug works very well for the lung cancer patient who carries the genetic mutation. Likewise, in myeloma, it also works.
That means that drugs that work on one type of cancer could work against another seemingly very different cancer because they share the same mutations. Which brings us back to Mahmoud. Turns out his myeloma shares a gene mutation with a skin cancer, malignant melanoma, and the same drug is effective against both cancers.
I'm doing good. There's no pain. Right now the lab results are very, very good. In a short period of time, the lab results are very good.
Cancer genetic profiles in some cases also allows doctors to know which drugs are unlikely to work against a given cancer, sparing a patient expense and side effects.
This individualizes care, basically personalizes it to a patient's disease and helps us direct the treatment very rapidly by repurposing the drug for cancer.
So will all this lead to a cure for cancer? Well, not in the same way that antibiotics cure infections, but it could easily turn cancer into a chronic treatable disease rather than a lethal one, and that could happen very soon. For Science and You, I'm Dr. Max Gomez.
Autoimmune diseases occur when our immune system, which should protect us, instead starts to attack healthy cells. For those who suffer from such diseases like lupus and rheumatoid arthritis, the effects can be debilitating. So what role does genetic makeup play? Tinabeth Pina has that story.
There are days when I literally have trouble doing things. When I don't, it's like a great day.
Carol Rhodes has lupus and APS, two of about 100 autoimmune diseases that affect over 20 million Americans.
First of all, the immune system is a very, very complicated system. It's as complicated as the brain, and its business in life is to recognize all those things in the outside world from which you should be protected, and at the same time, not attack yourself, which is also a complicated world.
When your immune system attacks the very organs it was designed to protect, it can make whatever is ailing you very difficult to diagnose.
There are so many vague symptoms, and it's the piecing together. And I didn't have all of the symptoms. I just had some of them. And it took about a year, or maybe 2 years, till they said, we think this is it, let's start treating it.
Carol has spent the last decade treating her lupus and APS, even though doctors can't tell her exactly why she developed them. She has no family history of autoimmunity, but lupus and APS, along with other autoimmune diseases like rheumatoid arthritis, alopecia, and vitiligo, can have a genetic component.
Genes can explain roughly 50%, maybe more, of the fact that you have an autoimmune disease. If you have rheumatoid arthritis, for example, or lupus, the rate at which you will have a sibling with those diseases is somewhere around 5%. If you have an identical twin with lupus, you have about a 30% of chance of getting lupus yourself, not 100%. So there are other things besides the shared genes that are contributing to this, and that has to do with running into the right environmental influence at the right time, and this kind of background of chance activity that goes on in the body.
Some environmental triggers of autoimmune diseases include exposure to viruses, smoking, and diet. And according to the National Institute of Health, autoimmunity is a major health concern for women. Do race and gender play a role in who contracts an autoimmune disease?
Race and gender is very interesting. Most autoimmune diseases are more common in women, although there are exceptions to that. Probably has to do with hormonal regulation of the immune system. The ethnicity is also interesting. So, and that's an interaction of environment and genes. So a really great example of this is African Americans. African Americans are at higher risk for lupus. When they get lupus, it tends to be more severe.
With the disparities among ethnic groups and genders, to the obstacles encountered in diagnosis and treatment, the diverse array of autoimmune diseases has made it difficult to find cures, but research may eventually change that.
There are lots of new treatments coming on board, and the reason that there are is because of advances in genetics and immunology. We really understand a lot more about the immune system now than we did even a decade ago. Where large numbers of families are being collected to try to find immune or inflammatory changes that are present in unaffected relatives of people with autoimmunity, with the idea that you could predict who's going to develop the disease. Those studies are not yet concluded, but you may actually be able to tell somebody, you have a 50 or 80% chance of developing an autoimmune disease within the next 3 years, and therefore we think you should take this drug to prevent it.
Remember, if you're diagnosed with an autoimmune disease, it's important to seek out the right specialist so you can get the exact treatment you need. For Science and You, I'm Tinabeth Pina.
Autism is a difficult and heartbreaking fact of life for many families, but scientists are coming together with groundbreaking genetic research that could make a real difference. Here's Donna Hanover.
So I believe that genetics accounts for about 80% of autism risk.
Dr. Joseph Bauxbaum, director of the Seaver Autism Center at the Mount Sinai Hospital, says in some cases the genetic source of autism is fairly simple, in others it's complex.
One of the things we found pretty quickly is that there are some genes that they are sufficient to cause autism, that are really, really red flags. That gene mutation is going to be associated with very high risk for autism. But in most of the cases, it's genes interacting together, what we call an additive effect. Gene A, gene B, and gene C add together to produce the risk for autism. We have good data that there are about 1,000 genes that contribute to autism risk. We have about 100 of those pretty well nailed down, and about 200 which we're getting quite sure about.
Studying DNA from families who have a child with autism has been vital.
These spontaneous mutations, you can only find them by looking at mom and dad and seeing that they don't have that gene variant. It happened when the sperm or the egg was formed. So mom and dad don't show that genetic mutation, but the child does.
Huge changes have taken place recently that help this research.
Technology over the past 5 and 10 years has completely changed everything we can do with genetics. So just a few years ago, we could maybe get information on a few genes per person. You and I have about 22,000 different genes. With the new technology, we can literally, over the course of a day, get information on all 22,000 of those genes in anybody who walks in the door.
How do genes cause autism?
So what we're finding among these top 100 to 200 genes is that many of them affect the synapse, or the point of contact between nerve cells in the brain.
Another way some genes cause autism is in flawed transcription regulation, the way they regulate when, where, and how much other genes are expressed.
So you have 22,000 genes on your chromosomes, and most of the time they're turned off. You don't need hemoglobin expressed in your brain. You need hemoglobin expressed in your bloodstream. So the gene for hemoglobin is going to be turned off in your brain. But we're beginning to find that there are mutations in genes that are critical in that process of turning genes on and off. We can pinpoint that in time to early development of the fetus in the first and second trimester, when these genes which are responsible for turning other genes on and off are most active.
Some autism gene mutations have been duplicated in mice to study further, especially for drugs that might help. And recently, scientists have been able to take skin cells from people with autism, turn them into stem cells, and then turn them into brain cells, all in a petri dish, where drugs can be tested on them in hopes of finding a treatment.
This is what families are hoping for, that you will find this. How close are you to finding some kinds of drugs that might help intervene?
So in the case of genes where the mutation is very high risk for autism, we're actually doing pretty well. For every one of those genes, there are clinical trials right now going on with novel therapeutic approaches. We certainly don't know how many are going to work and how well they're going to work, but this is such a different place. A few years ago, there was nothing to talk about. Now suddenly there are several dozen trials like this.
These are trials on people?
On people.
Every year, about 100 to 200 families give blood to get tested at the Seaver Autism Center. And starting in 2010, Dr. Bauxbaum helped create something remarkable, the Autism Sequencing Consortium, which got NIH funding in 2013. About 150 scientists from various institutions have agreed to share their data without waiting to publish so that they all have a lot more data quickly to speed up the process of figuring out what's behind autism.
We came up with a plan that we would all share our data prospectively and work together to analyze the largest possible samples as quickly as possible. There's no question that the Autism Sequencing Consortium has precipitated enormous progress.
Dr. Bauxbaum says the Autism Sequencing Consortium has collected more than 20,000 samples from families and expects to double that in another 2 years.
We already know that behavioral interventions are very effective in autism, especially when they're done early. So the more we can do about earlier and earlier diagnosis, which includes genetic testing, the better we can deploy behavioral treatments that will actually improve the trajectory of the child. For the higher functioning kids, we already can move them off the spectrum. I am very, very optimistic that what we're doing right now about understanding the causes of autism and how that manifests in the brain are going to lead to new treatments in the very short order.
I'm Donna Hanover for Science and You.
It's common for people to say, you have your mother's eyes, or you look just like your father. What you don't often hear, you got that disease from your mother or father. But perhaps we shouldn't be surprised. Magalie Laguerre-Wilkinson tells us why.
Advanced research in genetics has helped uncover many mysteries behind the causes of certain illnesses or conditions. Once a taboo subject, the research into which group is more prone to which disease is now not so taboo. Dr. Harry Ostrer, geneticist and professor of pathology and pediatrics at Albert Einstein, explains.
Every group has a significant genetic burden. And so, for instance, with some of the recent work that I've been doing at Einstein, we've been sequencing the genomes of people from different ethnic groups, Puerto Ricans, Dominicans, African Americans, Ashkenazi Jews. And we find that each of these groups has a significant burden, and in fact, there can be a reasonable overlap with regard to the diseases or the disease susceptibilities that may be found between one group and another.
One of the reasons behind this, Dr. Ostrer explains, is that once upon a time, societies weren't mobile. Their sedentary lives allowed for intermarriage. That phenomenon contributed to a host of specific conditions within specific groups.
Looking at a place like Europe, the genetic map of Europe really tends to overlay pretty well with the geographic map of Europe, so that there are sufficient differences between people in Ireland and Greece, or between people in Poland and Spain, that by analyzing enough genetic markers, one can really put them into the genetic map.
The neurological disorder known as Tay-Sachs disease is one condition that was once only considered to be found among Ashkenazi Jews. Research has revealed more.
There are common mutations actually in other groups as well. Some Jewish and some non-Jewish. So Iraqi Jews, Moroccan Jews.
Sephardic.
Have Sephardic Jews have Tay-Sachs disease, but in each of these groups, it's on the basis of their own private founder mutations. And that's true also for French Canadians and for Irish. So there are multiple different population groups in which there are common Tay-Sachs disease founder mutations.
So can early screening for expectant parents possibly stop the Tay-Sachs gene from being passed on to the next generation?
So yes, something can be done. With any of these conditions, of course, couples may be screened prior to pregnancy or during pregnancy. They may decide to have prenatal diagnosis. They may even decide to have in vitro fertilization and pre-implantation genetic diagnosis. So actually, a cell from the embryo is removed and tested to see if the embryo
is prone to
prone to have a particular condition, and then only those embryos that are thought to be normal will be implanted during the fetal fertilization. Certainly, that has had the impact of markedly decreasing the frequency of Tay-Sachs disease in the United States, as an example. And one of the remarkable effects of the screening programs has been that Tay-Sachs disease has gone from being a predominantly Jewish disease to being a predominantly non-Jewish disease.
Mainstream.
Yeah, because people who aren't Jewish aren't typically screened for Tay-Sachs disease.
So as research continues to try to solve genetic mysteries, the next phase is the Precision Medicine Initiative commissioned by the Obama administration, whose goal is to recruit 1 million people into a genetic bank in order to understand the genetic basis for diseases. Dr. Ostrer hopes his institution, Einstein, will be one of the sites to take in the information from candidates.
We think it's important that we become a site because, of course, we have this wonderful population in the Bronx, sure. And genetic studies should really be performed on, everyone and not just on, certain groups. So,
and there's pretty much everyone in the Bronx.
There is pretty much everyone in the Bronx. That is true. The Census Bureau says that the Bronx is the most diverse county in the United States.
Is that right?
That's what they say, which means the likelihood that any two people are related in the Bronx is going to be lower than it is in any other county.
I'm Magalie Laguerre-Wilkinson for Science and You.
Finally today, Lisa Beth Kovitz takes a very personal look at promising genetic treatments in muscular dystrophy.
In 1996, my friend Star and I each gave birth to wonderful, amazing, gorgeous baby boys, Jonah and Hurricane. As they grew, the two boys became great friends, inseparable in all activities. By the time they turned 5, though, Star noticed small differences in the boys.
Jonah would grab a bunch of toys and go running up the stairs while Cain had to go step by step holding onto the rail.
On May 5, 2001, he was diagnosed with muscular dystrophy, Duchenne's, the most aggressive form of the disease. Boys with Duchenne's rarely live into adulthood. But recent advancements in genetic treatment may be changing those odds. I invited Hurricane and Star to join me on this interview to get some clarity on the latest science.
Why is Duchenne's muscular dystrophy so aggressive?
Duchenne muscular dystrophy is caused by mutations in the dystrophin gene. And the dystrophin gene makes a really essential protein for the function of muscle fibers. And so when that muscle protein is missing, the surface of the muscle fiber can't contract the way it's supposed to, and it develops a lot of holes and leaks and breaks that the cell just can't repair.
What is it on the genetic level that's making this happen?
There are several different types of mutations in the dystrophin gene. The most common one that leads to Duchenne muscular dystrophy is actually the deletion of a middle fragment of the gene itself. So it's missing what we call exons, which are the little building blocks of the gene itself. And it's really important the way those little building blocks come together. They have to get assembled in the right order, and they have to have ends that basically match. And if those ends don't match, then the cell knows something's wrong with that molecule and it degrades it.
I read in the New York Times about genetic advancements in Duchenne muscular dystrophy. Can you explain that to me?
Absolutely. In these 3 back-to-back papers in Science, 3 independent groups used a brand new technology called CRISPR-Cas9 to do what we call gene editing. This takes advantage of an enzyme and what's called a guide RNA to basically direct that enzyme to the area of DNA that you want to destroy. And so that protein, guided by those RNAs, went to the abnormal section of the dystrophin gene in mice and basically lopped it out, forcing the cell to put the pieces of the DNA back together, repairing the gene.
In gene therapy, when they say cut, we're not talking about knives and scissors. How are things cut in gene therapy?
The gene editing, gene cutting, takes place using an enzyme that basically acts like Pac-Man. So it scans along the DNA till it finds the region that it's supposed to cut, and then it basically just snips through the 2 strands of the DNA. And then there are repair mechanisms that every cell uses to repair DNA, because this happens on its own at times. And so those repair enzymes come along, detect the break, and try to glue it back together. And if you've cut out the abnormal piece and it floats away, then the remaining pieces can basically be stitched back together by those enzymes.
Once your genes have been corrected, are you good for life?
That's the theory. So once you've spliced out, cut out the abnormal region and the chromosomes have been stitched back together, then with every cell division thereafter, that piece should still be missing and would still be missing. The problem for this particular technology is getting enough cells edited to make a difference.
How would it get from mice to someone like me?
The steps, as I see it, between what we just read about in the science papers and coming to people are, first of all, improving the efficiency of the gene editing. I think we need to see more muscle fibers expressing dystrophin and expressing higher levels of dystrophin to know whether it's going to work in people. Secondly, I think we have to make sure that those enzymes that get introduced are only cutting where we think they're cutting. And then we have to figure out how to safely deliver the virus that brings with it the enzyme package to a wider number of muscles and at a level where the enzyme gets turned on and can do its gene editing.
Most parents who have children with DMD, our biggest question is always, is that going to be available in his lifetime?
Duchenne's has gone from fatality in your late teens to lots of individuals living into their late 20s, early 30s. You know, we don't know what the upper limit is with steroids and good cardiac care and pulmonary care. So I think that it used to be that we were willing to take a lot of risks because the mortality was so young and so early. Nowadays, most boys have time. They become men, they get jobs, go to college, do all sorts of things that we never envisioned for kids with Duchenne. And now we have to, as a downside to that, take a step back and say, okay, we've got to find safe and effective therapies, not just try the kitchen sink even before it's ready and before we know that it's safe. But I think everyone in the field is more enthusiastic and optimistic than they've ever been about a treatment strategy or an approach. You know, 3 simultaneously published papers coming from 3 laboratories tackling this issue all at the same time reminds me that there are lots of very smart people working on this as fast as they can, right?
This has been Lisa Beth Kovitz for Science and You.
I'm Carol Anne Riddell. Thanks for joining us for Science and You.